Let's look at the cellular interaction with collagen fibrils in this module. Cells interact with extracellular matrix mechanically and chemically. We have said ECM, extracellular matrix, is basically a scaffold on which cells can rest. So there's a mechanical component and there's a chemical component. For example, these ECM molecules can provide signals to the cells directly, or they can sequester signaling molecules and act as a reservoir. Ultimately, they can have a major effect on the architecture of the tissue. Fibroblasts, which produce collagen, they work on the collagen they produce. They play with this collagen, they crawl on it, they tug on it, they pull on it, and they can also compact collagen molecules into sheets or cables. If we put fibroblasts on gel collagen gel, which is in random orientation, what happens is this collagen, which is the molecules are hanging out in random. These fibroblasts, they draw collagen, they pull the collagen in from their surrounding, and they basically result in a contraction of this collagen gel from its original or initial volume. Similar activities of a cluster of fibroblasts surround itself with a capsule of densely packed, circumferentially oriented collagen fibers. So, bunch of fibroblasts, they can grab collagen and they can form a capsule of this molecule around themselves. So, we will see that also that certain cells actually have a coating of collagen, especially type 4 collagen, around themselves. So, we will see that later. But here I would like to show you a photograph. Here you have explants. Explants is basically small tissue which is full of cells. So, a piece of, for example, chicken heart is placed on this side and also on this side, and there is collagen matrix in between. As you can see, initially the collagen matrix was basically at random. They have pulled this collagen matrix into this sort of a sheet, like a rubber band sheet structure. So this basically took about 4 days, and these two cells, they reoriented the collagen which is present into this sheet-like structure. And also, after they altered the collagen, some of these cells actually came out of the explant and they started crawling and growing on the collagen. So it is a two-way relationship. Cells can orient collagen molecules, and then collagen itself can serve as a track or as a substrate on which cells can move, or they can move along these tracks, and they can also use this molecule as a signaling entity which tells them how to or where to move. The fibroblasts, again, influence the alignment of collagen fibers, and in turn, collagen fibers affect the distribution of fibroblasts. Initially, they were just present in those explants we saw in the micrograph, but after the collagen has been oriented properly, the cells start moving on the newly oriented collagen substrate. Fibroblasts can generate long-range order in extracellular matrix inside the body to create tendons, ligaments, and also tough, dense layers of connective tissue that also can ensheath and bind most of the organs. So what we have seen, there is a two-way relationship between collagen and the cells, fibroblasts. First of all, fibroblasts secrete collagen. Then they play with the collagen, they pull, they form different shapes, they form long cables, or they can form sheets of collagen molecules. And in turn, this collagen molecule can affect the behavior of the cells. For example, in our experiment, the cells which were residing in those explant tissue, they came out of the tissue and started crawling on the collagen matrix that they had oriented.